So today our focus is going to be on cellular level, which is the second levels of organization. And we will focus first on the structure referred to as plasma membrane, which sets up the boundary for the cell, and also focus on how molecules have the ability to pass through this membrane through mechanisms such as active or passive transport. Now, to start with, we are looking at something referred to as cell theory, developed by Robert Hooke. And basically, it sets up a set of standards for what we consider to be a cell. So we consider cell the smallest unit of living organism that has the ability to carry out its own homeostasis for its survival. It has the ability to undergo cell division, or division, which we refer to it as mitosis, to create new cells. And it also has the ability to carry out normal physiological functions for its survival. If we take a look at the cells that are found within our tissues and within our organs, we can identify about 200 different cells that are different in not only their size but also shape, as well as their components that are present within them. You can see in the picture some of the main categories of the cell that we will be discussing in your class. These include your red blood cells, referred to as erythrocytes, the two different types of muscle cells, such as a smooth and a skeletal muscle cell, cells that are involved in creating electrical impulses, such as your nerve cells, cells storing fat, which we refer to as adipose, or basically fat cells, cells of immune cells such as macrophages, and cells of reproductive system such as a sperm. And by just looking at these cells, you can see the differences that exist based on their shape and size. Now, when we talk about cell, the definition of the cell, or easy way to identify or describe a cell, would be the smallest functional unit of a living organism. Now, when we look at a cell, we can break it down to three basic parts. The plasma membrane, which is basically sets up the boundary for your cell, and you can see it as this line going around. Cytoplasm, which is a space within the cell that contains its organelles. So basically, this space you see surrounding this, all of these structures are basically located within the cytoplasm. And the purple structure you see in the drawing, referred to as the nucleus, which contains the genetic information in the form of DNA, and is also referred to as the control center for the cell. Now, before we get into details about your cell, we need to understand that we have a specific vocabulary that describes the environment within the cell and the environment outside the cell. The word extra implies outside, and intra means within. So when I use the word extracellular fluid, or ECF, basically we're referring to the fluid outside your body and outside your cell, and intracellular fluid is referring to the fluid that is found within the cell. Going back to the extracellular fluid, or ECF, there are two main categories. Basically, the fluid that flows within your blood vessels, which we refer to it as blood plasma, and fluid that is found between the cells, that is referred to as interstitial fluid, or IF. So taking a look at this picture, what you see going around is basically the boundary of your cells, or boundary of your body, basically your skin. You can see your respiratory system up here. This is your cardiovascular system that is allow you to pump blood using your heart. You see these basically dots representing all of your cells within your body. Digestive system on the side, and what you see at the bottom of the picture represents your urinary system. So keeping that in mind, we are going to use this diagram and the color coding of the diagram to describe what is considered as extra versus intracellular fluid. So taking a look at the first picture we have, basically you understand that about 60 to 70% of your body is made up of water. So, and you can see it based on this picture that the total body water is a significant amount of space. So, this picture highlights what we consider as extracellular and intracellular fluid. So basically anything that is found in your blood, inside the cell, and the environment that surrounds your cell. Now, intracellular fluid, you can see that is referring to the fluid that are found in your cells that are highlighted in blue. And this also applies to red blood cells that are found inside your plasma, because red blood cell, as the name cell implies, that is still identified cells. This could also refer to your white blood cells that are also circulating within your bloodstream. Now, this picture, the third picture in this scenario, represents extracellular fluid. So if I take a look at the first picture, and I basically subtract the second picture, which is my ICF, whatever I'm left with would be my ECF, or my extracellular fluid. Now, extracellular fluid, as I mentioned, can divide to plasma, which is the fluid that is flowing inside your blood. So this is blood vessels, and you see it blue, basically highlighted in blue. And anything that's remaining, which is basically the structure that surrounds your cells, surrounds your organs, that is referred to as the interstitial fluid, or IF or ISF. This gives you a better idea about the volumes of waters that you have in your body in liter. I don't require you guys to know these numbers, but it gives you an idea of what these volumes are, that again makes majority of your body mass. Now we are going to start talking about the first component of your cell, which we refer to as plasma membrane, which sets up the boundary for your cell. So if you take a look at the plasma membrane is made up of phospholipid bilayer. As the name implies, it is made up of phosphate and lipids in a form of fatty acid chains. And the word bilayer implies that it is two layers. So 75% of your plasma membrane will be phospholipid. Looking at the characteristics of the phospholipid, you have the phosphate heads and you have the fatty acid tails, which are the lipids. Now, phosphate heads have polar characteristics. They are charged and they love to interact with water, so water loving. And as a result, we use the word hydrophilic to describe their characteristics. The fatty acid tails is the lipid part of your phospholipid. These are non-polar, which means there is no charge. And as the word lipid implies, they do not like to interact with water, so they are considered to be water-hating or hydrophobic, as in they have a phobia, they're afraid of water. So if you take a look at the membrane, the circles that you see in this picture are representing your phosphate heads on either side, bilayer, and the line coming off on either end would be representing the fatty acids with hydrophobic characteristics. About 5% of your plasma membrane is glycolipids. The word glyco implies sugar, and lipid still forms the lipids. So these are basically lipids that are bound to a sugar component. And about 20% are a specific form of lipid, which we refer to it as cholesterol, to increase the membrane stability and make sure the cell is maintaining its boundary. Now, taking a look at your plasma membrane or your phospholipid bilayer, embedded within that, you also have membrane proteins. These membrane proteins are also essential for the functionality of the cell. And these proteins as well as the phospholipid bilayer molecules have the ability to move around within the plasma membrane, and it is creating a very fluid structure for your plasma membrane, which we refer to it as fluid mosaic pattern. Now, if I want to make sure that my cells stay attached to one another, depending on their function and their location, I can connect them or allow them to communicate with each other using a specific structure referred to as cell junctions. So before we go further and talk about cell junctions, let's take a look at membrane proteins that are embedded or present or associated with your plasma membrane. The first category of these molecules are these proteins are referred to as integral proteins. Integral remind me of the word integrated or inserted within the plasma membrane. So these proteins, as you see them as these green structures expanding through the phospholipid bilayer, will be passing through the bilayer and having access to inside and outside your cell. Now notice that these proteins are interacting with both the phosphate heads and the fatty acid tails. Considering the characteristics of the phosphate and the fatty acid tails, these integral proteins will have a very similar characteristics. What that means is that the outer edges that are interacting with phosphate, and you see them in green, right over here and right in this region, these parts of protein will have hydrophilic characteristics as they are close to both the phosphate heads as well as a watery environment. The central region, which is interacting with the fatty acids, are also going to carry characteristics similar to fatty acids, which are considered to be hydrophobic or lipophilic, as in they like to interact with fat, lipophilic, but do not like to interact with water, hydrophobic. Now, these proteins usually act as a transport protein for your cells, and we'll discuss them in more detail as we talk about the transport mechanisms of your plasma membrane. Now, the second categories of the proteins are referred to as peripheral proteins. These are loosely attached to your integral proteins. They can be found on the outer edge as well as the inner aspect of your cell. These proteins typically have hydrophilic characteristics since they are directly associated with the phosphate heads as well as the watery environment of either ECF or ICF. Typically, they act as a cell-to-cell connection, and in some cases, they also act as enzymes. Please do keep in mind that what I'm talking about when I talk about integral protein and their functions and peripheral proteins and their functions, these are some of the examples of functions associated with these cells. They will, of course, carry other functions that are not described in this PowerPoint. Now, as I mentioned, cells are interacting with each other and are communicating and are connected together using a structure called cell junctions. However, again, there is exceptions to the rule where cells have no connections or junctions with their neighboring cells, such as your blood cells and sperm cells. There are three different junctions that are present within your cell. The first one is known as tight junction, second one is desmosomes, and third one is gap junctions. Let's take a look at each of these cell junctions and some of their characteristics and an example or two of where you can find them. Tight junction, which is represented right here, is a way that where you have interlocking proteins connecting the plasma membranes of two cells together tightly. This allows for a very impermeable junction that almost limits the movement of majority of the molecule to limits the movement of majority of the protein within this space. Now, that being said, there's always again exception to the rule, and that is something we'll discuss in more detail for the urinary system. But tight junction, and as example of a tight junction, would be your stomach cells that are lining basically the stomach cavity. These cells must protect the underlying tissue of the stomach from the acidic environment of the cavity produced by hydrochloric acid, and therefore all of the cells are connected via tight junction. The second type of junctions we have is desmosomes. Desmosomes are very similar to zippers, as you have these proteins are coming together, and basically from one cell to another cell they fit into each other to form these linker proteins, or to form these linkage between each other and ensure that basically the cells are remaining attached with each other. Another example or another way you can think about them, think about Velcro. So the desmosomes are very similar to Velcro structures that connect the cells together, and these structures actually expand throughout the cell as well. So these extensions you see right here continue to become the linker proteins you see between the two cells. Desmosomes are extremely important for anchoring your cells together. These are essential for cells that are undergoing significant amount of pressure, and you want to ensure that these cells will not fall apart under the significant pressure. For instance, in your heart, you have cardiac cells that are connected together via desmosomes and a structure known as intercalated disc. This is to ensure that as your heart pumps, that the force is divided evenly and the pressure is not placed on specific cells, ensuring that your heart basically can maintain a significant amount of pressure for a long period of time without damaging the cell. Lastly, we have gap junctions, and as you can see, and as the name implies, these are proteins that are expand through the membranes of both cells, allowing electrolytes as well as ions and molecules, ions to pass from one cell to another. This is really important for cell-to-cell communication. As we discussed in the case of cardiac cells, gap junction is a mechanism that allows your cardiac cells to spread the electrical impulses from one cell to its neighboring cell. And I'm going to stop this PowerPoint for now, and we'll pick up the rest of the information in the second part of this lecture.